everyone, my name is Rebecca Gonzales-Leon, and I will be doing my presentation for you all on cat scratch disease. I hope you enjoy it. Here is a quick overview of the different topics I will be discussing. Cat scratch disease, the organism, the history, the epidemiology, the transmission, disease as it presents in humans, prevention, and control. Okay, let's start with cat scratch disease. Cat scratch disease, CSD, is also called cat scratch fever or Bartonella Hensley infection. It is a bacterial infection that is spread by cats. The disease spreads when an infected cat licks a person's open wound, bites, or scratches a person hard enough to break the skin surface. While domestic or feral cats have been found to be carriers of this disease, kittens actually have a higher transmission rate. The organism. Cat scratch disease is a bacterial infection that is caused by the Bartonella Hensley bacteria. Cats are the natural reservoirs for the Bartonella Hensley. The bacterium may be present in 3 to 68% of even healthy cats and is more common in younger animals, strays, and those with fleas. Fleas are the main vector of transmission, and here you can see this is a flea, adult size. Bartonella Hensley is an aerobic, oxidase negative, and slow-growing gram-negative rod that is slightly curved, as shows here. And you can appreciate the shape. Here is the rod, slightly curved. And here is an example right here of the gram-negative stain. Now, the average size of the bacterium is 2 micrometers in length by 0.5 to 0.6 micrometers in width. Like other Bartonella species, it can grow on chocolate agar or Colombian agar supplemented with 5% sheep or rabbit's blood. On to the history. Now, symptoms similar to cat scratch disease were first described by Henry Harnaud in 1889. However, a clinical syndrome likely to be cat scratch disease was first reported in 1950 by Robert Debris. In 1983, the Warthin starry silver stain was used to discover a gram-negative bacillus, which was named Amphibia felis. Then in 1988, we see the organism was successfully isolated and cultured. In 1991, the CSD bacillus was named Amphibia felis after the Armed Forces Institute of Pathology, where the organism was actually discovered. That takes us to 1992. An organism known as Gonzales-Leon Hensley was isolated from an HIV-infected patients with bacillary angiomatosis, pelvis hepatis, and fever syndromes. This study and others refuted the role of Amphibia felis in CSD in favor of Gonzales-Leon species. In 1993, the genera Bartonella and Gonzales-Leon were united. Thus, Bartonella Hensley is currently recognized today as the causative agent of cat scratch disease. That brings us to the epidemiology. The epidemiology, worldwide distribution. However, there's a prevalence in warm and humid climates. 22,000 to 24,000 new cases are reported annually within the United States. This is actually more common among children. Serial prevalence in domestic cats is 14 to 55% and 30% in wild feral cats within the United States. In the United States, 28% of surveyed cats actually showed having antibodies against the organism. In California, blood cultures were positive in 56% of domestic cats that were younger than 1 year old, and in 34% of cats older than 1 year old. Bartonella hensleyi is endemic and in Europe, Africa, Australia, and in Japan. The transmission. And here we have a lovely picture that was given by the CDC and located on their website. And it shows the steps of transmission. Right here we show in step 1, the eggs shed by the female. And they go into the environment. The eggs hatch into the larvae. The larvae then form pupae. And then from there, the adults hatch from the pupae. And here we can see a full adult flea as the main vector. And then he will go on to infect the cat or kitten. And then a human will play with their cat or kitten, and either their skin will break via being bitten or scratched, or saliva will be transferred onto the human, and that is how they will contract cat scratch disease. And we will go a little bit more in depth with that next. Okay, so continuation of the transmission of the disease. After a cat becomes infected, the bacterium typically lasts anywhere from 5 to 8 months, but sometimes it may last for actually over a year. After clearing of the strain of Bartonella hensleyi, cats may be infected again with different types of the strain. It is transmitted through cat scratches and bites, or to the exposure of the cat saliva. The lesion is usually small, about less than 1 centimeter in length, and the infected area may appear swollen and red with round raised lesions and may contain pus. Now, in a person, signs of cat scratch disease typically occur within 1 to 2 weeks of being exposed to the bacterium. The disease as it presents in humans. Symptoms. Usually this is mild and self-limiting, except for towards immunocompetent people. Now, they may experience more severe symptoms, and we will go into that a little bit more in depth next. 90% of patients with cat scratch disease will actually develop bumps, papule, or blisters, pustules, at the site of the injury that appear just like this one, a reddish brown in color. Usually now they're non-tender to touch, non-tender. Usually the first sign of this will occur 3 to 10 days after being infected. And other symptoms might include some fatigue. Fever has been reported in some patients. And if so, they usually report it lasting several days. Also a headache, as well as lymph node swelling and pain near the scratch or bite. This actually occurs 1 to 4 weeks after the infection. The swelling may last for 2 to 4 months, and sometimes pus may form inside of the lymph nodes. There's an overall feeling of discomfort and malaise from this disease. And here we have a more visual picture for you all to appreciate with regards to the symptoms. And we see here the site of the wound. And isolaterally, same side right here, you would see swelling in the lymph nodes. Right here too. In some circumstances, there is an enlargement of the spleen, such as shown here. And again, right here, there's a bite or a scratch or a wound. And if you follow that down, same side, right underneath the armpit here, you will see another swelling of the lymph node. Okay. Now, for the immunocompromised individuals, such as those that have AIDS or even younger children, symptoms can be worse. Now, they might exhibit larger bumps, such as this one right here, or this one down here. And also lymph node swelling. Now, the lesions could also spread wider, and sometimes the bumps can even appear on their eyes, such as this one right here, causing a condition called paranoid ocular glandular syndrome. Inflammation of the brain can also occur, which would then cause seizures. Once again, those are rare cases. Cat scratch disease diagnosis. Cat scratch disease may be diagnosed presumptively in patients with typical signs and symptoms and a compatible exposure history. Serology can confirm the diagnosis, although cross-reactivity may limit interpretation in some circumstances. Lymph node aspiration is not recommended, only except to relieve pain and severe pain and swelling, or in cases where the diagnosis is deemed unclear. B. Hensley DNA may be detected by PCR or culture of lymph node aspirates or blood, though sensitivity of these methods is not optimal for the blood samples. Since B. Hensley is a fastaceous, slow-growing gram-bacterium, cultures should be held for a minimum of 21 days. Cat scratch disease and treatment. Now, I already mentioned before, and I will mention again, you usually do not need treatment. Usually it's self-limiting and people do heal. However, in cases where it's severe, antibiotics have been deemed helpful. And in that event, if that were to occur, studies have shown that azithromycin led to a more rapid resolution of lymphadenopathy than placebo. Now, in cases with complication, for example, the azithromycin, the Z-Pak, 500 milligram daily for 1 week would be taken, and then it would be followed by an additional 250 milligram for 4 weeks as part of continued treatment. Okay, on to prevention and control. Prevention and control. Now, this depends on avoiding bites and scratches from cats, particularly kittens and strays. Cat owners are advised to avoid any type of rough play with the cats in order to reduce their risks of getting scratched or bitten, or the transference of saliva onto a person. Any wounds inflicted by that cat should be washed and disinfected immediately and thoroughly. Allowing the cat to lick the skin, particularly any open wounds, is always discouraged. Nail clipping is also important. Declawing does not appear to affect the transmission, but keeping the nails clipped is highly suggested. Flea control, of course. Flea control may also be a preventative measure since the fleas are the known vectors. However, this is not even certain. Vaccine information. To date, there's currently no vaccine that is available for cat scratch disease. However, if one were to contract cat scratch disease, that would actually give them immunity for life. And this concludes my presentation on cat scratch disease. I hope you all enjoyed it. Thank you and have a wonderful day.